Hola a todos, bienvenidos a la segunda presentación del día. Para comentarios como nota, se va a hacer en inglés para que todos pongan atención. Entonces, comenzamos. Federico Fries received the bachelor's degree in electrical engineering from the Zurich University of Applied Sciences in 2016 and completed his master's in science degree in rehabilitation engineering and assistive technologies from University College London in 2020. He has experience in working at the Singapore University of Technology and Design as a full-time research assistant for over 3 years. He has interest and focus on the development of sensory devices for different applications such as robotics. So I'm going to leave you with Federico, and I hope that you enjoy his presentation and hard work through all the year.
Hello, first, thank you for the introduction, and also thank you to the UCL Mexican Society for organizing this event. It's a great opportunity for us to share our work. And well, also thank you to the ones that managed to connect themselves to this lecture. What I'm going to talk about is about my project during the master's dissertation, which was the development of a wireless pH measuring device for orthopedic implants. A little bit of background about this topic. Musculoskeletal problems is one of the most widespread health problems. This happens because during the lifetime, joints and bones are exposed to high degrees of wear. So after some point, they stop working as they used to. So people start having pains in the hip, knee, ankles, and they have trouble moving. So these problems can be cured through treatment, but also another common procedure is to replace the damaged bone or joint with an orthopedic implant. In the UK alone, around 2.5 million orthopedic implant surgeries were done in the past 15 years, and this involves hip, knee, ankle, arm, wrist, and so on. The problem is that 15% of these replacements failed due to poor osseointegration, which is the integration between the implant and the bone, or due to infection. And what happens when the implant fails is that basically it has to be replaced by another one. So the patient has to go through surgery once more. And you can imagine at 80 or 70 years old going through surgery in such period of times is not ideal. So this problem cost the NHS, the health system in the UK, from 7 to 11 million pounds a year, and similar statistics can be found in the US, Australia, Germany, etc. A little bit about the project. The outline is going to be, I'm going to talk about how implant fail due to infection, how does this infection come to place. Then it was established that there's a strong relationship between bacteria and pH. This is why we're doing a pH device. I'm going to explain the specifications of the pH sensor, how this sensor has to be for this application. And I'm going to talk a little bit about the available technologies for pH sensing and why I chose the sensor that I chose for this project. And finally, I'm going to show you a little bit about the implementation, so you can have an idea of the result of the end device, how it looks like, and so on. Bacterial infection. How does this bacterial infection come to place on orthopedic implants? This can happen from the time you implant the implant in the body until like forever, as long as it's there. But the most critical time is the first 3 months after implantation. This is because there is no integration between the implant and the bone. So what happens is, if a bacteria manages to go into the body, they can adhere to the implant surface. As you can see here on the image, there's the implant, and here the bacteria adheres to the implant. Then it starts maturing and formation, and you can see here these blue things that is covering the bacteria. This is called a biofilm, and this one protects the bacteria. So once this biofilm is formed, it is very hard to eliminate the bacteria. Not even antibiotics manage to go through this biofilm and kill it. And afterwards, the bacteria starts dispersing. This can happen in only 6 hours after the bacteria attaches to the implant. So the most common bacteria is Staphylococcus, specifically Staphylococcus aureus. And a way to prevent this is through material science. Scientists have developed coatings to coat the implant and avoid or prevent this addition from bacteria. There are also other ones that they can even release antibiotics once they detect a bacteria attached to the implant. However, these materials are very hard to produce and very expensive. Therefore, they are not in the market yet. So this shows us how important it is to have a tool that diagnoses this bacteria as soon as possible. And by as soon as possible, I mean a few hours. The treatment would be antibiotics. If detected early, it can work. If not, then the implant has to be replaced. And in the worst case, there is an amputation of the limb. It was established that there's a very strong relationship between Staphylococcus and pH. In general, there are a lot of physiological processes in the body that are pH dependent, like infection, tumor growth, wound healing, etc. So this tells us that pH is a potential indicator for bacterial infection. So I did quite a extensive research about these topics, trying to understand how can the infection be identified through the pH. Many researchers have done a lot of work in this area, but some of the main conclusions are that pH levels are above 70 means that there is an infection. Maybe some of you don't know the pH scale, which goes from 1 to 14, and 7 would be a neutral state. So if we have levels above that, then we have an infection. Also, that Staphylococcus aureus is more prevalent at higher pH levels. And addition of the Staphylococcus aureus to the implant can be prevented if we have lower pH levels. Nonetheless, there are a lot of results, there are a lot of discussions about this topic. And the truth is that no one has ever measured how the pH should behave after the implant is implanted. So we don't really know for sure how the pH looks when we have a healthy recovery process. We don't know. We also don't know how it looks like when there is an infection, and especially we don't know how it looks like when we have a Staphylococcus bacteria infection. There are other bacteria, of course. We also don't know that. So this shows the relevance of having a device capable of measuring this pH at the surface of the implant. So what is the idea of this entire project? The idea is that we have here a bone that is fractured, and we use a fixation plate to repair this fracture, right? In this case, it's the wrist. And the idea is that we have a circuit or electronic circuit that is integrated here into the plate. And there are sensors that measure the pH attached to this fixation plate. One of the characteristics of this device is that it's wireless. That means that it can send the information measured to an external device, which could be a phone, like the ones we have, or a tablet. That makes a lot easier for the patient and also for the doctors to measure and monitor the recovery process. So I'm going to use a pH sensor, right? That's established. And how does this one have to be? Based on the research that I did previously, I figured that the pH range should be between 4 and 5. It's more or less the range where infections happen and also the healthy recovery process. The size should be around 200 square millimeters, which is like around this. And this is to fit into this fixation plate. I chose this fixation plate because it's a very small one, and after this we can actually use we can actually integrate this PCB into many other implants. Has to be high sensitive. That means that very little changes in pH should be measurable. The lifetime should be at least 3 months. Like I said before, the crucial times is the first 3 months after implantation. Low drift. Drift is an error that happens. Many electronic devices and sensors have this error. And this is basically, if you have, for instance, a drift error of 0.1 pH per hour, that means that if you are measuring a solution with a 7 pH, after 1 hour you have 7.2, then 7.1, sorry, then 7.2, 7.3, and so on. So imagine if you measure for 3 months, then you'll have a huge error and it saturates the sensor. The device should be able to power through NFC. NFC is a technology that all of us have in our cell phones. Many of you have these cell phones that can charge wirelessly. So this is NFC. It works through electromagnetic induction. And the nice thing about this is that at the same time, it can transfer the data measured by the sensor. Finally, it has to be biocompatible. This is to ensure the safety of the patient and that there are no other problems caused by this device. What are the technologies to measure pH? To this day, I only found 2 different technologies that are used to measure the pH levels at implant surface. These are these 2 images that you see on the top right corner. The first one uses hydrogels, which are elastic materials based on water, and they can have different characteristics. And what this happens is that when the pH changes, this hydrogel swells or shrinks. So this swelling and shrinking can be measured through plain tomography. Maybe you cannot see it probably in this image, but you can see it showed how after 60 minutes, the pH falls and the size of the hydrogel change. Another option would be capacitive pH sensors that have electrical characteristics, and they change their electrical characteristics thanks to the pH. This can be measured through electrical capacitance tomography. The problem with these 2 technologies is that both require imaging instruments, which are only available at hospitals and clinics. That means that you as a patient, you have to go there, take the image, has to be processed, and then you can know, okay, maybe I have an infection or not. So what we want to have is something that is integrated into the implant and can give you information without you having to go to the hospital. Because as you see, it's very fast. It takes only a few hours to have this infection. So if you're the patient and after you're going home after the surgery, you can be monitoring yourself if there is a problem, if the pH levels are okay or not. So I did some research to find which would be the best sensor. There are many other technologies that can be used, but we wanted something that does electrical signals. And I chose potentiometric systems, which are basically composed of 2 components, which is a working electrode and a reference electrode. And there is a potential between both of them. When the pH changes, the potential changes between both, and that potential is measured. The working electrode would be an iridium oxide sensor. The research showed that these sensors are the best sensors for this application. Why? Because they fulfill all the parameters that I discussed in the previous slide. They have low drift, high sensitivity, wide pH, fast response, they're durable. And the one that I chose in this case was one proposed by Nguyen in 2013. And the reason for this is because it is flexible. Why is this important? Because implants have very odd shapes. So if you have a stiff sensor, it is hard to integrate into the implant. So if you have a flexible sensor, this could be a lot easier. And here's an image of this sensor on the corner. You see they were working in the reference electrode and the film over here. This is very small sensor. This area is 2 by 2 millimeters, so it also allows you to integrate almost everywhere. Finally, a little bit about the implementation. I don't want to tell a lot about this for two reasons. One is too complicated, and I would need a lot of slides to explain this. And the second is because I have submitted a few papers, and these results are going to be published in a couple of weeks. So I don't want to share too much about these details. Nonetheless, I want to share you how this looks and the end product of this thesis. Basically, the architectures of these implants have a sensor, which is the iridium oxide sensor. Then we have a microcontroller that processes this information, this signal from the sensor, and then transfers it into a transponder, which is, everything is actually encased in this integrated circuit, the blue one, the green one. And these transponders, the only thing that it does is translates the information into a protocol that is later sent through the antenna to the external device, which is the cell phone. We have in this case here, this blue box is a circuit that helps minimize the error measured by the iridium oxide sensors. These are very sensitive sensors. There's a lot of noise in the body, you have to imagine. And this box helps you minimize this error and increase the accuracy of the device. Once we have this block diagram, we can design the system, we can design the circuit, and this is how it looks like. It's, as you can see, it's 11 millimeters by around 15 millimeters. It's very small. Here are the two components on the top and the bottom, and here are these paths to connect the iridium oxide sensors. The iridium oxide sensors have to be fabricated separately, and then everything is integrated into the implant. So this, I came so far, this can be produced now. I haven't done that, but I will be doing that in a few days. So to sum up my entire talk, musculoskeletal problems are widespread everywhere. It's very common, and that's why we have a lot of implants implanted every year, everywhere. However, there's a high implant failure rate, and this is because of the bacteria that manages to get into the body. And this tells us how important it is to have a tool that can help us diagnose these infections, because in the end, we're gonna save a lot of money. The patient is gonna be much safer, only has to go through surgery once, and it's better for everyone this way. There's a strong relationship between pH and Staphylococcus aureus. This is why we chose pH to measure the infection. And this research allows us to establish the specifications of the device. And finally, I chose these iridium oxide sensors because they fulfill all the specifications that we discussed before, and I showed the implementation. So this is the end of my talk. I hope you could understand. If not, I'm happy to answer any questions. Here's my email. I also have a webpage here. You can contact me through that. And yeah, thank you very much for listening.
All right, Federico, thank you very much for your talk. It's a very interesting approach to a very difficult problem. Definitely, infections by several bacteria are a hard problem to tackle, most in this current age of antibiotic-resistant bacteria. So I have one question, like, or two questions.
Okay, sure.
Right. So first, could this idea be applied to monitor other bacteria of medical interest like Pseudomonas? And how difficult is to work with such small components, and how quickly or efficient in terms of cost could this approach be applied to, for example, countries where medical installations are not widespread, like, for example, some countries in Latin America or Africa, right?
Yes. So the first question, I know that infection, like I said, there's a relationship between bacteria and Staphylococcus and pH. It is possible to measure other kinds of bacteria. However, there's not a lot in this context because most of the infections happen because of Staphylococcus. And if it's Staphylococcus, it can be aureus or epidermis. Obviously, there are other kind of bacteria, but they are less aggressive as well. Therefore, we focused on this one. I'm not sure if pH would be the best solution to measure other bacteria, but it would definitely be possible with this. The second question was how hard it is to work with these kind of components. Well, this device, I would send it for fabrication, and the components and everything would be fabricated together. So I wouldn't have to do any of the soldering. It's very hard to do this by hand. Possible, but it's a lot easier if you get the provider to do it for you. They have machines, they do it in the oven and everything. It's a lot better. However, the connection between the iridium sensor and the PCB and also the attachment to the implant, these are problems obviously that are there. I didn't explore those ones because I didn't have the prototype, so there was no point on actually working on it. But it's definitely a challenge. It's definitely a challenge to find the right material to attach the sensor, to attach the device. We also have to package the entire device so that it's biocompatible and the patient is safe. So it's a difficult problem. There are many problems that come one after another, but I think this is a start. Once we have the device, we can test it, and once we know that it works, it measures reliable and everything, then we can go to these questions that you asked. And finally, about the cost, yes, this is a problem. Iridium is very expensive, so I know that this problem is not just for orthopedic implants. It can happen for any other implants, such as pacemakers, even catheters, screws for bones, whatever, right? For instance, I once talked to a cardiologist, and he told me that pacemakers have the same problem. But the thing is, for instance, pacemakers cost 300 pounds, and the device alone would cost around 300 pounds alone. So the device gets a lot more expensive. However, like I said, if there is a bacteria, the only solution, if you don't detect it early, the only solution is to remove the implant. So it has to go through surgery, has to go through rehabilitation. The patient has to stay at the hospital again a couple of days. So in places like the UK, that the government or the NHS pays for these services, or in Mexico, the IMSS or ISTE, right, that has to be paid by the government, it means that the patient has to be back at the hospital. So maybe long term, the entire process would be cheaper if we do it this way rather than focusing on the cost of the implant itself.
Yeah. Okay, so we have another question from Luis Fernando. What is the expected durability of the device or sensors that you are proposing?
Yes, very good question. This was a very important parameter that we measured. Ideally, it has to last for 3 months because it's the most critical time. It's the time it takes for the osseointegration. Most sensors do not last that long because these are chemical sensors, so there is a loss of electrons and all these kind of things. So most sensors do not last that long. The one that we chose does. That one lasts for months. Unfortunately, because I did a literature review, most of the researchers didn't study how long these sensors survive. So I don't really know for sure how much all these sensors would work. Ideally, the project was meant to be done in the lab, and I was supposed to fabricate these sensors and measure these parameters. But hopefully there is a sensor that can last at least 3 months, if possible even longer, because infection can happen even after these 3 months as well. But critical is the first 3 months.
Okay, we have a complementary question from Luis Fernando too.
Okay. Yes, that's the so the question, that's the idea. Hopefully, yes, that we can reduce the infection rate. I mean, infection can still occur. I mean, the bacteria can still attach to the implant. However, the key is to detect it early, right? To prevent. And then if you know that there's an infection, you can start subministrating antibiotics, and then you can eradicate the bacteria.
Yeah. Like definitely, like with any infection, the idea is to catch it up early, right, in the early stages, so people will have a better outcome of survival
Yes,
than if it's caught like in the later stages. And
exactly.
I agree with you that the cost of certain components are certainly a challenge to manufacture devices, right? But I guess that's why we're here and trying to find ways
Yes.
to manufacture and develop new technologies in a cheap way to make people, be able to obtain them. Okay, Federico, like if you can tell us a bit about your experience at both UCL and the UK during your postgrad.
Yes, I really enjoyed my time at UCL as long as it lasted. Then COVID happened and we had to go home. But I really enjoyed it. I was based in Stanmore, which is the Royal National Orthopaedic Hospital, one of the best orthopedic hospitals in the world. And our program was very practical, so we had a lot of projects. It was very hands-on, and the projects came directly from the hospital, so it was very nice to be able to work on these kinds of projects because we were really involved. We also had a lot of people from the hospital, like doctors, clinicians, nurses, rehabilitation specialists, people that do assistive devices, everything. They come to us to give us lectures and teach us. And it was very small groups, so you can talk to them, you have discussions, everything. So overall, I really enjoyed my time at UCL. I really like my program, which is Assistive Technologies and Rehabilitation Engineering. Definitely recommend going to UCL. London as well is a great city. There are a lot of things to do. And if with the support of the Mexican Society and the advice that you guys gave us at the beginning, it was possible for us to find cheap ways to do stuff and have a bit more fun and enjoy the city a bit more.
Yeah, well, that's the idea, right? The society is the hub for any Mexican student that wants to approach and have a community, right? A community in which you can, have a good time. Also, like if you have any doubts or inquiries about the life here at UCL and the UK, it's the main hub that you can go to. So please be uh, sure that we can make you and help you enjoy your stay here at UCL in London.
Yeah,
And
you did
Federico,
a great job.
yeah, thank you very much. Yeah,
Thanks.
and Federico, thank you very much for your talk. I'm sure that we all wish you the best for your publication and for your next project. And
Thank you.
we enjoyed your talk very much. And well, if just as a closing remark, thank you for accompanying us during this second round of talks. And if you want to present your work or want to present something, remember that we're going to have our talk about sessions in the coming term.
Sure.
So feel free to attend us by the virtual talks that we're going to be allocating in the next month. So thank you very
Okay,
much.
great.
Have a good afternoon and
You
have a
too.
nice week too.
Thank you. See you.
See ya.